So following glycolysis, the link reaction, and Krebs cycle, we've got reduced NAD and reduced FAD from Krebs cycle. And remember, the Krebs cycle is taking place inside the mitochondrial matrix. And so what we're going to do next is consider what's referred to as oxidative phosphorylation, which involves the electron transport chain and a process called chemiosmosis. So we do need to appreciate the structure here, because you need to make sure that you have this, as I said, is a mantra constantly, an idea of how structure and function are related. So we've got the outer membrane, and the outer membrane is impenetrable to hydrogen ions. So no hydrogen ions can pass through this outer membrane. The inner membrane is very, very folded. It's folded into what's called cristae, and the inner membrane is folded to increase the surface area. And within this membrane here, we've got many, many, many embedded proteins, some of which are electron carriers, and electron carriers, as the name suggests, are allowing electrons to move. It's called the electron transport chain, because electrons move from one protein to the next protein to the next protein, and as they move along, they're moving down an energy gradient. And as they move along and move down the energy gradient, what we're going to do is release free energy. Glycolysis produces reduced NAD. The link reaction produces reduced NAD. Krebs cycle produces both reduced NAD and FAD, and these reduced forms of the cofactors are then passed into the electron transport chain and chemiosmosis. So this is the part that we're going to cover next. These two together are referred to as oxidative phosphorylation. And what we need to appreciate is that the Krebs cycle, which is producing most of the reduced cofactors, is doing this inside the mitochondrial matrix. So we've got lots of reduced NAD that are found in the matrix. And here we've got a diagram. The matrix is inside here. So the matrix is got many, many chemicals in here which allow it to do its job, which is the enzymes of Krebs cycle. And it's also got lots of NAD, which are ready to be reduced into reduced NAD. So the mitochondrial matrix is here. We also need to think about the structure and the function and its relationship for the other parts too. So the outer membrane, this outer membrane here, you need to appreciate, is impenetrable, impervious to hydrogen ions. So hydrogen ions can't go out and hydrogen ions cannot go in. The inner membrane is folded into these cristae, and the cristae are going to increase the surface area of that inner membrane. What's also interesting and important about the inner membrane is that the proteins which are floating around, remember, make the connection with the fluid mosaic model. The mosaic referencing this idea that we've got proteins which are embedded in that phospholipid bilayer. So the proteins in here are they're numerous, and some of the proteins are called electron carriers. And electron carriers are forming the so-called electron transport chain. And what we do in the electron transport chain is we take an electron and we pass that electron from one carrier to the next carrier to the next carrier. While we're passing it along, what we're doing, remember, is reducing. When we take the electron away and give it to the next one, we've got a redox process. When we add an electron, we're doing reduction. When we remove that electron, we're doing oxidation. And what we're going to do as we pass that electron down the electron transport chain is that the electron will move down a so-called redox potential. And that means that you are going to generate free energy. Essentially, the electron starts off being very, very highly energetic. And as it's moving along, it's going to be at a lower energy level. And the differential between those energies allows free energy to be useful, which we'll come to in just a moment. So within this inner membrane, we've got electron carriers. Also within this inner membrane, we've got pumps. And remember that pumps are involved in active transport. And these active transport pumps are pumping hydrogen ions. So within here, we are pumping hydrogen ions, and we've got electrons jumping about. We've also got a channel which is allowing facilitated diffusion of hydrogen ions. So remember that we've got pumps which are doing active transport. Active transport is moving molecules up a concentration gradient. So we're going to move hydrogen ions, which are here in the matrix. We're going to pump them up into this inner mitochondrial space. And what we're then going to do is we're going to allow them to diffuse back, but because they're diffusing down an electrochemical gradient, they're going to diffuse back through different forms of proteins, which are called carrier molecules or channels. So the channel, which is allowing for facilitated diffusion, is also associated with another protein, which is ATPase, an enzyme. So this inner membrane here, the folded cristae, is rich in electron carriers, rich in proton pumps, rich in proton ion channels, and also rich in ATPase, all of which is allowing us to do the two parts that are the electron transport chain and chemiosmosis and oxidative phosphorylation. The other thing to bear in mind is that the membrane space here is very small, which means that as we pump the hydrogen ions in here, it's relatively easy for us to build up a concentration gradient of hydrogen ions. Now, because hydrogen ions are ions, they've got a positive charge, I'm not going to refer to this as just a concentration gradient. I'm going to refer to it as an electrochemical gradient because we've got a high charge here and a high concentration of hydrogen ions here and a lower concentration or charge down here in the matrix. So structure function is super important. This again is taken from BioNinja, and I think this covers it visually quite nicely. So let's start with the reduced NADH here. Now what we're going to do is we're going to reoxidize the reduced form of NAD, and we're going to take that hydrogen ion away from it. That allows NAD then to be used again in the link reaction, a Krebs cycle, etc., etc. But this hydrogen is then busted into a hydrogen ion, a proton, and also an electron. Now the electron, which is coming from the hydrogen, is then going to be passed along these electron carriers, and as it's going along the electron carriers, it's losing energy, and that energy which is being lost as they move along the electron transport chain, I'm going to call free energy, and that free energy is allowing the pump to operate. And here what we can see is the pump, and the pump is moving hydrogen ions, protons, through the inner mitochondrial membrane and into the intermembranous space. So what we're going to do is take hydrogen, we're going to break the hydrogen into a proton and electron. The electron is going to move through the electron transport chain, create free energy that allows the pump to pump protons into the intermembranous space. So what we then generate is a very, very high concentration in the intermembranous space of hydrogen ions or protons. That high concentration of protons in here is very, very important. So we're generating a so-called electrochemical gradient across this inner mitochondrial membrane. This next diagram is showing you the same thing. And again, these PowerPoints are in here. So it's talking about the space, and it's talking about taking this hydrogen, moving the electron and moving the proton. So the proton is pumped, and the pump is operated by energy which comes from the electrons moving down the electron transport chain. And again, we can see this visually in these diagrams here. So what we've got is a very, very high concentration of protons within the intermembranous space. And the next step is that we are then going to allow those hydrogen ions to be controlled and diffuse down through this channel. So we've got facilitated diffusion going down an energy gradient, high concentration of protons, lower concentration of protons. So as the hydrogens are moving down through this carrier protein, this facilitated diffusion, we call this a proton motive force. The proton, hydrogen ion, is moving and creating a flux because it's a charged ion. So we got a proton motive force as we move down through here. So the first energy I'm going to call free energy. The free energy generated from the electron transport chain is going to pump the hydrogen, and then what we've got is this hydrogen ion flux, this proton motive force, that's allowing the energy that's required to take ADP and add the phosphate to create ATP. So the phosphorylation process is taking place in combination with the channel protein, and also here surrounding we've got ATPase. So ATPase and the movement of hydrogen ions through it, the proton motive force generated allows you to rephosphorylize the ADP into ATP. And This next diagram is showing you the same thing. So we need to now consider, well, if the hydrogen ion is coming back through here, what happens to the hydrogen ion? Also, if the electrons are jumping around, what happens to the electrons? So the electron here is at the end of the electron transport chain. The hydrogen ion, which is diffusing back, is then going to combine with a molecule of oxygen. So the electron, the proton, combines with oxygen to form water. So we call oxygen the final electron acceptor. Without oxygen, we cannot do this last stage. And without oxygen, we can't do Link, Krebs, and all of this oxidative phosphorylation. So this last stage is called oxidative phosphorylation because it's using oxygen and it's combining that with a phosphorylation reaction. But it depends on the action of the, first of all, the electron transport chain and the pumping of, the pumping of the hydrogen ions. Sorry, wrong slide, this one here. So it depends on the electron transport chain and the pumping of hydrogen ions. That's all to do with the electron transport chain. This part here is referred to as chemiosmosis. This is the movement of hydrogen down its electrochemical gradient. And then we've got the final stage where oxygen is the final electron acceptor, and we've got oxidative phosphorylation. So again, chemiosmosis and electron transport chain, they're happening, both of these processes are going on at exactly the same time. Together, collectively, they're called oxidative phosphorylation, but obviously the two depend on one another. So these slides also you can find in the classroom, and again, they're showing you visually the same thing, slightly different images. This slide here is just reminding you of what a paradigm shift is. That's a shift in an accepted form of thinking. And it was in 1961 that Peter Mitchell proposed this idea of a chemiosmotic effect. And that was replacing the idea that came before it. You don't have to know the details of that, but you can find out more if you wish by looking at the link down here. We've got a summary. Again, this is in the Google Classroom. This comes from Stephen Payne. This one is just reminding you that you've got to make sure that you link structure and function. This could come up as an essay question. How is the mitochondria structure allowing it to do its function? So you talk about the outside membrane. The outside membrane is not penetrable to hydrogen ions, which allows the high concentration to be generated here. You've got the electron carriers, you've got the ATPase, etc., etc., etc. So just make sure that you map that out very carefully. And again, there's a summary of everything that I've said for you to take a look at here. There's a couple of past paper questions. So have a look at this, see if you can answer it, pause the video. Okay, and there's the answer. Similarly, here's the question. This is the kind of thing that you might expect on an exam. Pause the video, see if you can do it. There's the answer. Okay, so you could be asked to annotate, and you could be asked to connect the structure and the function. And this is a really nice summary here. BioNinja covers this really nicely. iBiology might be worth a look at, and obviously go back to your big fat textbook as well, and Cognity. So I'm going to ask you to now work through the booklet, make sure that each part of that booklet is clear in your mind. I would also make additional notes, and summary mind maps are really very, very powerful way of putting all of this together. Talk out, there's a lot of terminology, and this does come up quite often as an essay question. So just make sure that you are very, very clear with the language and the processes here.